Good afternoon, everyone, and welcome to this edition of the Aquaculture webinar series. This series is brought to you by the National Aquaculture Association, the North Central Regional Aquaculture Center, and the United States Aquaculture Society, and is designed to enhance aquaculture production and educational opportunities throughout the United States. Today's webinar is entitled Antibiotic Resistance in Aquaculture. Know the risk, know the facts, and will be presented by Dr. Jesse Trzynski with the Idaho Department of Fish and Game, based just outside of Boise. She supervises the Eagle Fish Health Laboratory, which provides comprehensive fish health services to IDFG and its partners. Prior to joining IDFG, Jessie was an associate professor at Southern Illinois University, where she managed one of the leading fish nutrition groups in the country. Jessie is currently president-elect of the American Fishery Society and will begin her presidency in 2018. Thank you for being with us today, Jessie. Take it away.
Thanks, Alan, for that introduction, and good afternoon, everyone. The subject of antimicrobial resistance and the creation of superbugs that are resistant to treatment with currently available antibiotics has been a hot topic for some time, but the subject has been particularly prominent as of late. There was much discussion in the months leading up to and since the rule change that took effect at the beginning of 2017, making all antibiotics accessible by veterinary prescription or veterinary feed directive only. In fact, this was the primary justification for putting all antibiotics under veterinary oversight, to ensure these drugs are being used legally and judiciously and not in such ways to contribute unduly to the development of antibiotic resistance. As we will discuss in this webinar, there are legitimate risks associated with antibiotic use or misuse, and antibiotic resistance is a legitimate concern for the treatment of disease in humans and in animals. That said, it's important to understand that legal and judicious use of antibiotics is an acceptable practice, and approved antibiotics can be used in such a way that does not jeopardize animals, people, or our ability to treat disease in the future. One bit of housekeeping for those who are viewing this webinar live. If you have questions, you can ask those using the Zoom Q&A function. I'll try to answer as many of those as I can at the end as time allows. But feel free to enter those questions into the queue at any time during the presentation. Now, before I get into the nitty-gritty, I want to note that I'm going to be sharing data from a number of different projects in this presentation. I want to acknowledge a number of people and institutions that have been involved in that work. Most of these individuals are or were associated with my old lab at Southern Illinois University, or they're currently a member of the Eagle Fish Health Laboratory, part of the Idaho Department of Fish and Game. But Jim Bowker with the U.S. Fish and Wildlife Service has also been involved, as well as Matt Aardsma at Purdue University. The work that I'm presenting here today wouldn't have gotten done without them, so thanks to everyone listed here. First, let's start with the basics. What is antibiotic resistance and how does it develop? Microbes are eternally mired in an arms race, developing new and exciting ways to destroy each other and to avoid being destroyed. In the broadest sense, this is what antibiotics are: substances secreted by bacteria or fungi to kill or inhibit the growth of other competing microbes. Although this form of microbial warfare was well known, it wasn't until Alexander Fleming started working with what he called mold juice, ultimately discovering penicillin. It was after that that the therapeutic value of these substances was ultimately harnessed. Acquiring factors that will allow a microbe to persist in the face of another's biochemical onslaught is a natural and an adaptive process, but there are a number of ways in which we can influence that process with potentially dangerous results. This handy figure from the Centers for Disease Control illustrates how human-influenced antimicrobial resistance happens. In any population of bacteria, a few individuals will be naturally resistant to certain types of antimicrobial molecules. When that particular molecule is applied, it kills most, but not all of the bacteria. The ones with the resistance factors are spared. These then go on to proliferate, such that the population becomes dominated by the resistant bacteria over time. Now, there's nothing too complex here. This is just natural selection and survival of the fittest in the context of a petri dish. But what makes things a little bit more complicated is the means by which bacteria like to trade bits of their genetic material among individuals and across species. Through these genetic swaps, resistance factors can find their way from completely innocuous bacteria to those that are important animal or human pathogens, making treatment of these diseases more difficult, or in some cases, impossible. Now, in aquaculture, there are a number of practices or conditions that can make resistance more likely. And the first element is biosecurity. We do our best, but when you're dealing with water, especially in the outdoor environment where there can be predators or weather events, different factors that cause water or fish to move, it's difficult to ensure complete biosecurity and keep bacteria completely isolated. Many farms and hatcheries have multiple production lots, different stocks, or different age classes on station at any time. Now, as fish age, like us, they become more resistant to different types of bacteria. They develop immunities against them after they're exposed and ultimately survive. When young fish encounter older fish, they can pick up infections that the older fish have developed an immunity against. Now, whether it's because of regular introductions of these younger, naive hosts, or because you have a pathogen in your water source that you can't do much about, it's pretty common for farms and hatcheries to encounter the same pathogens with some regularity. If you don't have many treatment options and you have limited ability to avoid or eliminate a pathogen altogether, this might increase the odds that you're going to be battling the same bacteria more than once and potentially encountering resistance at some point. Much of this is true of human medicine and certainly terrestrial animal health management, but water and connectivity in aquaculture do add a bit of complexity. This is another helpful infographic from the CDC that helps to contextualize antibiotic resistance in animal agriculture. As before, we have a population of bacteria, some of which are naturally resistant to an antibiotic, and these will proliferate and come to dominate the population following antibiotic treatment. The only difference is that these bacteria have either infected that animal or they're part of the livestock's normal flora. If the bacteria are pathogenic, repeated exposure to the same antibiotic can make treatment less effective over time. Now, whether they are important animal pathogens or not, these now resistant bacteria can come into contact with humans and human pathogens in a number of ways, including consumption of contaminated animal products or produce, poor food handling practices, or by coming into direct contact with the animals or with their waste. Now, there's a very good argument that antibiotic use or misuse in human medicine is a much more significant contributor to human health risks and antimicrobial resistance at large. But drug use in animal agriculture, including aquaculture, has been linked to the development of antibiotic resistance. That's not necessarily human health concerns, but at least the development of resistance in the bacteria themselves. There are potential human health risks to consider, but from the agriculture perspective, there is also the more immediate and practical issue of being able to maintain antibiotic effectiveness for as long as possible and to keep as many drugs in the medicine chest as we can. There are very few antibiotics that have been approved for use in U.S. aquaculture, and it's important that we maintain their functionality and effectiveness for as long as possible, given that it usually takes decades and many millions of dollars to get a new drug through the approval pipeline. In terms of human health, when we talk about antibiotic resistance in fish, we're not necessarily concerned about the direct human health risks of resistance developing in fish pathogens. There are very few bacteria that are pathogenic in fish and in humans. But what can happen is that fish pathogens can come into contact with other bacteria that are pathogenic in people. Through those genetic swaps that I mentioned earlier, it's possible for fish bacteria to share resistance factors with a human pathogen, leading indirectly to resistant infections that can cause illness or death. Again, we're several steps removed in aquaculture, so this isn't as likely as direct resistance development due to antibiotic use in human medicine, but it is possible. Okay, so we don't want resistance to develop because it's a fish health management concern and a potential human health concern. So how do we know when it's happening? The first thing that you might consider is treatment effectiveness. If I apply an antibiotic treatment to my fish and it doesn't work, does that mean that the bacteria are resistant? Well, maybe they are, but maybe they aren't. As it turns out, treatment effectiveness is a very poor predictor of resistance because there are a number of different reasons why a treatment might fail that don't have anything to do with antibiotic resistance. First, was the treatment appropriate? Are we talking about a pathogen that's susceptible to antibiotics at all? For example, you can treat a viral infection with antibiotics all day long, and it won't make any difference because antibiotics don't affect viruses. Second, was the treatment applied in a timely fashion? There comes a point when an infection is too advanced for any antibiotic to make a difference, and if you wait too long, your fish may be beyond the point of no return. Finally, was an effective dose delivered? Did you get enough medicated feed into the fish, and did you treat long enough for the medication to work? This is somewhat related to timely treatment because if you wait too long, the fish might go off feed, but can also be related to things like water temperature or other factors that influence feed consumption. These are just a few reasons that an antibiotic treatment might result in effective control of mortality, like the green curve, or treatment failure, like the red curve. And these have, again, nothing to do with antibiotic resistance. So how do we know if a bacteria is susceptible or resistant, and whether it's developing? One of the most common classical microbiological techniques to determine susceptibility is disc diffusion testing. The technique is really quite simple. First, a bacterial colony is isolated, either from a fish or some other type of sample, and then it's cultured on an agar plate, like the ones you see here. After the bacteria have been plated, a number of small discs impregnated with different antibiotics are pressed into the surface of the agar, allowing the molecules to diffuse out into the surrounding media. Now, as the bacteria grow, they'll form what's called a bacterial lawn over the entire plate, except where they encounter sufficient concentrations of antibiotics that curb their growth. If the bacteria are susceptible to the antibiotic, you will see a clear zone of inhibition surrounding the disc where the bacteria couldn't grow. If the bacteria grow right up to the edge of the disc, that tells you that the bacteria are not susceptible to that particular molecule. There are some important caveats to consider when comparing different bacteria or different antibiotics, but in general, the size of the zone of inhibition can tell you something. For example, if you see a small zone of inhibition, that tells you that the concentration of the antibiotic has to be pretty high to have an effect. Whereas if you see a large zone of inhibition, that tells you that the antibiotic can be effective in killing that bacteria or slowing its growth, even at very low concentrations. Disc diffusion testing doesn't tell you everything that you might want to know about antibiotic susceptibility, but it does give you very basic go, no-go information quickly and easily. And that can be really important when you're waiting on that diagnostic information to decide on a course of action to resolve mortality at a farm or at a hatchery. This is why the Eagle Fish Health Lab and many other diagnostic labs do disc diffusion testing on pretty much every bacterial pathogen that's isolated. Now, susceptibility, at least how we assess it in terms of disc diffusion, is not precisely the same thing as antibiotic resistance. Antibiotics work via a number of different modes of action. Some may disrupt the cell membrane or the bacterial cell wall. Others interfere with bacterial protein or DNA synthesis, etc. Bacteria can then counter these effects by modifying their cell walls or cell membranes, by using efflux pumps to get rid of the antibiotic molecules, or they may produce enzymes to inactivate them. Developing antibiotic resistance means acquiring the genetic means to activate these resistance mechanisms and defeat the antibiotics at their own game. Now, that's not exactly what we're measuring when we do disc diffusion testing. If bacteria have a negligible zone of inhibition, it's possible that they have acquired one or more of these resistance mechanisms. Or maybe they've always had it. Maybe they just aren't affected by that particular molecule. You can't say that for sure based on disc diffusion testing alone. But what we can say is that shifts in disc diffusion results and changes in zones of inhibition can suggest that the antimicrobial resistance factor is developing. I mentioned before that there are very few antibiotics that have been approved for use by the Food and Drug Administration for use in aquaculture in the United States. Most of you will probably recognize the approved drugs for use in aquaculture poster, which summarizes all of the FDA-approved drugs and claims for U.S. aquaculture. We're really only talking about a handful of drugs, and not all of the ones listed here are antibiotics. In fact, these are the only antibiotics that are approved, the products listed here in blue. In U.S. aquaculture, we're really only talking about a handful of ingredients: fluorphenicol, sulfodimethoxine or metaprim, tetracycline hydrochloride, and tetracycline dihydrate. These are the active ingredients in the approved antibiotic products. Other active ingredients like erythromycin are present in drug products that are currently in development and may be used under INAD exemptions. These include products like aeromycin as well as aquamycin. Within the Eagle Fish Health Lab, we are very concerned with protecting the effectiveness of this limited number of drugs, maintaining our ability to treat infections, and protecting human health in Idaho and certainly beyond. So we recently completed a study to answer this question. Is antibiotic resistance developing in Idaho Department of Fish and Game operated hatcheries? The Eaglefish Health Lab has a treasure trove of historical and current data describing bacteria we have isolated from various fish and game operated hatcheries. But with any data exploration exercise, you have to set some boundaries. We wanted to be able to test for temporal trends in antibiotic susceptibility. If you're only detecting a pathogen once in 20 years at a particular hatchery, that's not very helpful in examining the effects of time. Antibiotic resistance can develop in a few weeks, a few months, years, or decades, depending upon conditions. So we decided to restrict our data set to those facilities with isolations of a particular bacteria or causative agent in at least 10 consecutive or non-consecutive years. Our thinking was that if antibiotic resistance were happening, we should be able to see some sort of meaningful difference over the course of at least 10 years. After removing the data that didn't meet these conditions, we were left with records for 3 pathogens or diseases: cold water disease caused by Flavobacterium psychrophyllum, furunculosis caused by Aeromonas salmonicida, and motile Aeromonas septicemia caused by a number of different Aeromonad species. These were isolated from 12 different hatcheries, and you can see those that are listed here on the map, and they're spread throughout the state of Idaho. We tested these various isolates against 4 different antibiotics. You'll note that we use the active ingredients or proxies for the active ingredients for Aquaculture, terramycin, and Romet, as well as for the erythromycin products. Now, for convenience, in our lab, we test every bug against every drug. Now, of course, we wouldn't ever recommend treating cold water disease with a drug like erythromycin, for example, but historically, for convenience, we've tested all pathogens against all of the different antibiotics that we have available. So if nothing else, it provides some comparative information, as you'll see in just a moment. Once we amassed all this data, we worked with our in-house statistician to probe that data for indications of temporal trends in antibiotic susceptibility. I've given away the answer with the title of this slide, but let me take a moment to orient you to the graph so you can fully understand the meaning of this data. Along the x-axis, we have year of isolation, and along the y-axis, we have diameter of the zone of inhibition. The red line indicates the threshold of susceptibility. If the zone of inhibition is greater than about 18 millimeters in this case, the isolate is considered susceptible to fluorphenicol. If it falls below this line, it's considered not susceptible. Each of the dots represents an isolation from a particular hatchery in that year. Each of the black lines is a linear regression illustrating the predicted trend for a particular hatchery. So one of these lines might represent the predicted susceptibility of Flavobacterium psychrophyllum isolated from Hagerman State Fish Hatchery, whereas another line might represent the same from the Nampa Fish Hatchery. The first important takeaway is that the vast majority of all of the isolates fall above the red line. Since 1998, almost every isolate of Flavobacterium psychrophyllum is susceptible to fluorphenicol. The second takeaway is that none of the linear regressions that you see here are actually statistically significant predictions. The regression model that we developed was not able to reliably predict susceptibility from one year to the next, and that's actually a good thing. There is no significant trend, positive or negative, suggesting that the susceptibility of this bacterium to fluorphenicol is changing over time. It's just not. None of these lines are actually a statistically significant relationship, but if you wanted to argue that there could be something biologically meaningful here, regardless of the stats, most of these lines show an increasing trend or greater susceptibility over time. Now, there are a couple of lines in there that might suggest a slight, non-significant negative trend. If there are any pessimists listening to this webinar, you might be thinking, well, that's the evidence of declining susceptibility and antimicrobial resistance. Again, the stats don't really support that, but even if you think that relationship is real, keep this in mind. One of the facilities with the slight negative curve is the Eaglefish Hatchery, where Aquaculture has only been used once. The other line is for Haysper Fish Hatchery, where Aquaculture has never been used. So if you think that particular trend is real, and that's a very big and unsubstantiated if, there's little to nothing to suggest that use of Aquaculture at these facilities was a contributing factor to declining sensitivity to fluorphenicol. I'm going to breeze through the rest of the results pretty quickly. This is still Flavobacterium psychrophyllum, and down in the lower left panel are the fluorphenicol results that I just showed you. But now we've also got the same information for sulfadimethoxine or metaprim, tetracycline, and erythromycin. Flavobacterium psychrophyllum doesn't seem to have much susceptibility to the active ingredients of Romet, but the rest of the data look pretty similar. Most of the isolates fall above the susceptibility line, and none of the trends are statistically significant. So across the board for psychrophyllum, there are no trends in antibiotic susceptibility. Here are the results for Aeromonas species that cause modal Aeromonad septicemia, and it's a fairly similar story. Bear in mind that there are multiple species of Aeromonas involved, and species composition might be a factor here. We're actually going back through our data to tease some of this apart, but generally speaking, there's nothing here that suggests a change in antibiotic susceptibility over time. And then finally, we have our results for furunculosis. This is our smallest data set, so it's not quite as strong or definitive as the others, but the general implications are the same. Most isolates collected from 1998 to the present are susceptible, and there's no significant trends. Up in the upper right-hand panel, the tetracycline panel, this might seem like a pretty positive trend. The stats say otherwise. But even if this were a real pattern, it would suggest the Aeromonas salmonicida is actually getting more susceptible to tetracycline over time. So in terms of fish health management, that really isn't a concern. Not every active ingredient that we tested is appropriate or approved for use against the different target pathogens involved in this study. As I said, we test every bug against every drug simply for convenience. But that said, Flavobacterium psychrophyllum and Aeromonas salmonicida appear to be consistently susceptible to florfenicol and tetracycline also appears to be consistently effective against the causative agents of MAS. Now, does this mean that every treatment is completely successful in our hatcheries 100% of the time? No, of course not. As I explained before, there are a number of reasons why an antibiotic treatment might not work. Antibiotic resistance is one of them, but so is the severity of infection, the timing of treatment, whether fish are actively feeding, etc. Our data collectively indicate that some of the most important bacterial pathogens in our hatcheries remain susceptible to the antibiotics tested, and we see no evidence of meaningful change in susceptibility or resistance over time. Now that's very good news for Idaho Department of Fish and Game, but what kinds of practices could lead to antibiotic resistance? Initially, antibiotics were developed for therapeutic use in human medicine, but there are now a wide range of antibiotics developed and approved for use in animal agriculture, including aquaculture, as well as plant agriculture. It's this last one that you might not be as familiar with, but yes, some of the same antibiotics that are used in humans are also used in swine, poultry, cattle, fish, as well as different plants. In fact, these images represent a selection of the approved therapeutic uses of oxytetracycline-based products that are used to treat chronic bronchitis and other infections in people, a number of bacterial infections in these different animals, as well as turkeys and sheep, as well as bacterial infections in pears, apples, and a variety of other plant seedlings. Using antibiotics to treat something that isn't susceptible to them, either an unsusceptible bacteria or a non-bacterial pathogen, those are risk factors for antimicrobial resistance to develop. Similarly, not completing a course of antibiotic treatments is another risk factor. These are the common do's and don'ts of antibiotic treatments when you're using them according to one of the claims that are listed here. As I said, antibiotics were originally developed with therapeutic purposes in mind, with the idea of treating an infection in a sick person or a sick animal. However, it was discovered that treating healthy animals can, in some cases, enhance their growth performance or their growth efficiency. Naturally, commercial animal agriculture is always interested in ways to make livestock grow faster or more efficiently. So this incentive led to the development and approval of so-called production claims, or non-therapeutic uses of antibiotics. In most cases, these are the same antibiotics that are used therapeutically to treat infections. They're just applied at lower doses when used to promote growth. Again, to illustrate, here's some information about the different claims that were approved by the Food and Drug Administration for various oxytetracycline dihydrate products in pigs, chickens, cows, and some species of fish. For a specific therapeutic claim in chickens, you can medicate feed with oxytetracycline at a rate of 100 to 500 grams per ton, but the concentrations allowed for non-therapeutic production claims are a tenth of that. It's similar for cattle. Therapeutic claims allow you to apply OTC at 50 to 200 milligrams per head per day, but for production claims, the application rates are only 25 to 75 milligrams per head per day. It's a little different story for the claims for pigs, but generally speaking, non-therapeutic production claims are different from production claims in that therapeutic claims are high dose, short duration, and the production claims are low dose, long duration. It's important to note that the claims that are currently approved for oxytetracycline products in fish are for therapeutic purposes only. Oxytetracycline is not approved for any production purposes in fish, and in fact, there are no non-therapeutic production claims approved for any antibiotic in any species of fish. After the discovery of antibiotic-based growth promotion and the subsequent approval of various drugs for these different production claims, non-therapeutic use of antibiotics became quite widespread in U.S. livestock production. Of course, more recently, this practice has become quite controversial given concerns related to the use of antibiotics as a whole and whether certain use patterns, like these, can contribute to the development of these antibiotic-resistant superbugs. Now, as I said before, there are no antibiotics that can be used as growth promoters in fish in the United States, yet there's a pretty well-known misconception that antibiotics are used this way in hatcheries and farms. Hopefully, the people listening to this webinar know that drugs, antibiotics or otherwise, need to be applied judiciously and only to sick animals that need treatment. But the average person on the street only knows what they hear in the news or maybe on Facebook, and when it comes to aquaculture, that information is often deeply flawed. We know that using antibiotics to try to improve fish growth or efficiency is not only illegal, it's probably not going to work. I doubt that any of the producers that are on the line right now have ever observed really phenomenal fish growth right after treating an infection with an antibiotic. In fact, in most cases, it's all about getting the fish treated, healthy, and back on a normal feed as quickly as possible. That's how we think about improving growth and saving money. But there really isn't much information out there to suggest that antibiotics do or do not work as growth promoters in fish. Again, the public doesn't really understand this, and they are, by and large, quite skeptical of aquaculture. So simply telling them that the use of antibiotics as growth promoters in aquaculture is illegal doesn't necessarily do much to assuage their concerns. They might be thinking, well, if something is profitable enough, it doesn't really matter if it's illegal or not. Some people might find a way to do it. So given that there wasn't much information out there, we conducted a series of experiments to answer the question, does oxytetracycline promote growth or efficiency in fish? Our experimental design was pretty straightforward, involving only 3 dietary treatments: a control feed containing no oxytetracycline, feed medicated to achieve a standard therapeutic dose of oxytetracycline, and feed medicated to achieve a dose equivalent to about 20% of the standard rate. To create these feeds, we took a standard commercially available feed and we applied the OTC by top dressing, first with the antibiotic and then a small amount of fish oil to seal everything in. We didn't use the oxytetracycline in the control diet, of course, but we did top dress it with oil in the same way to keep everything consistent. The rates that we chose were intended to deliver 0, 80, or 16 milligrams per kilogram of fish per day if fed at 3% body weight per day. And that's, at least in terms of the standard dose treatment, in line with the current approved therapeutic use. It's really important that everyone listening understands that this study was not conducted in support of production claims for antibiotics in U.S. aquaculture. I don't think this sort of thing is a good idea for fish or any other types of livestock, and frankly, I didn't think it was going to work anyway. This experiment was all about determining whether antibiotic-based growth promotion is or is not a real phenomenon in fish. We ran the feeding trials essentially the same way that you would run a typical nutrition trial or growth trial. We took juvenile hybrid striped bass with a starting weight of about 27 grams, stocked them into a multi-tank recirculation system. We then randomly assigned each diet to 4 replicate tanks and then fed fish their assigned diets once daily at 3% body weight per day with with feeding rates adjusted for growth every 2 weeks. At the end of the trial, we counted and group weighed the fish to determine weight gain, specific growth rate, and feed conversion ratio. And we also euthanized 4 fish per tank for necropsy and a general fish health evaluation. Everything I just described was for a trial with hybrid striped bass, but we also conducted similar trials in channel catfish and Nile tilapia. The diets and experimental design were essentially identical for the other trials, except that these fish were different sizes at the start. Nile tilapia were stocked at 20 fish per tank instead of 10 fish per tank, and the water temperatures were a little different as appropriate for these different taxa. Okay, so let's dive into the results. The next few slides are going to be similar, so let me orient you to the figures. These data are the weight gain, feed conversion ratio, and specific growth rate for hybrid striped bass. The control diet is in gray, the standard or higher oxytetracycline dose feed is in blue, and the low dose is in brown. The p-values associated with the ANOVA test are at the top, and as you can see, there was no significant difference among the dietary treatments in weight gain, feed conversion ratio, or specific growth rate. The addition of oxytetracycline to these feeds at a low dose or a high dose did nothing in terms of hybrid striped bass growth. Here we have hepatosomatic index and viscerosomatic index for hybrid striped bass. VSI was unaffected, but we do see significantly higher HSI values among fish fed the low-dose OTC feed. Keep in mind that elevated HSI values are not desirable, either in terms of growth performance or dress out. And in fact, elevated HSI can be an indicator of liver dysfunction or toxicity. So these data would suggest that there's certainly nothing good about long-term feeding with oxytetracycline and potentially something pretty bad. On to the tilapia results. As with hybrid striped bass, we have no significant differences in growth performance. It's not statistically significant, but if there is any pattern here, it would be to suggest that feeding oxytetracycline long-term has an undesirable effect on weight gain, feed conversion ratio, and specific growth rate. One minor note, I didn't forget the error bars for FCR and SGR here. They're just too small to see at the current scaling for these figures. Hepatosomatic index and viscerosomatic index for Nile tilapia showed no significant differences based on treatment. Channel catfish, as we saw before, no significant effect of oxytetracycline on weight gain, FCR, or SGR. If there is a pattern here, it doesn't suggest anything positive about long-term feeding of oxytetracycline. And as we saw before, no significant differences in either HSI or VSI for channel catfish. Now, for the most part, we didn't observe the internal or external features of fish to be affected by dietary treatments. But we did see one significant difference in the appearance of skin and channel catfish. Among the control fish, we observed a number of abnormalities, but for the most part, these fish looked like typical channel cats. The abnormalities that we saw in the control group were not really severe. They were mostly things like light skin, blanched skin, attributes that could normally be associated with rearing these fish intensively in a recirculation system, especially one that has light-colored tanks. However, the frequency of abnormalities was significantly increased in both the low OTC group and the standard dose OTC group, with the majority of fish in both of these treatments having abnormal appearing skin and body surfaces. And in the case of the oxytetracycline groups, these abnormalities couldn't be readily attributed to what we typically see when you try to rear catfish in light-colored tanks. In these cases, we observe fish with reddened fins and minor hemorrhaging all over their bodies, as you can see in this picture here. So again, not something that suggests a positive effect, a growth-promoting effect of long-term feeding of oxytetracycline. So what does this all mean? Well, continuous long-term feeding of oxytetracycline at a standard therapeutic dose or a lower dose had no significant effect on growth performance of any of the fish we tested. The trends, when present, seem to suggest that if anything, long-term feeding with oxytetracycline has a negative effect on growth performance. So collectively, our results indicate that there's no benefit to long-term feeding of oxytetracycline, and therefore, there's essentially no incentive to misuse oxytetracycline this way. Legal or otherwise, why would a farmer pay more for medicated feed if it gave him equal or maybe even reduced fish performance? One thing to note is that since the beginning of the year, access to antibiotics has changed. All antibiotics, including products that were previously available over the counter at your local feed and farm supply store, are accessible now only by veterinary prescription or feed directive. In addition, the Food and Drug Administration is currently working with drug sponsors to ask them to voluntarily withdraw their non-therapeutic production claims for their products. So there's a lot of change going on. What are the current rules for antibiotic use? To address concerns related to the use of antibiotics in food-producing animals, and that includes fish, the Food and Drug Administration has issued a rule that took effect at the beginning of this year, and that changed how we access antibiotic products. As of January 1st of 2017, all immersion treatments, this would be water-based treatments with antibiotics, can only be applied under the authority of a veterinary prescription. As for the medicated feed treatments, those also require veterinary oversight, but this is called a veterinary feed directive. Some of this rule change has been confusing and has also created a few problems with respect to drug treatments in minor species. And so in anticipation of those issues, the Food and Drug Administration also released a new compliance guidance regarding extra label use of VFD drugs in minor species. And I'll talk a bit more about that in a moment. It's important to note that only antibiotic drugs were affected by this. So for aquaculture, for the drugs that are now available by prescription only, that's oxytetracycline. There's a variety of different commercial products available, but it's all that same active ingredient. These are the products that are used for skeletal marking, mostly in hatcheries. As for VFD drugs, these are the medicated feed treatments. Fluorphenicol, oxytetracycline dihydrate, and sulfadimethoxine or Metaprim are now available only by veterinary feed directive. Most of these are for specific therapeutic treatments, or in the case of oxytetracycline dihydrate for skeletal marking. Of course, there were a lot of drugs that weren't affected by this rule change. The January 1st rule does not apply to any other drugs, including other antimicrobials. This only applied to antibiotics. So things like chloramine T, formalin, hydrogen peroxide, and other related products were not affected by this rule, and they can still be accessed in the same way that they have been before. The rule also didn't affect any drugs that were accessed as investigational new animal drugs or INAD drugs. So anything that you have been using in coordination with the U.S. Fish and Wildlife Services ADAP program, those are currently unaffected. And that includes antibiotics that are INAD drugs. So generally speaking, antibiotics have to be accessed with veterinary access or veterinary oversight only, except for the antibiotics used through the INAD program. The rule change also didn't affect the deferred regulatory status drugs, like this is copper sulfate and potassium permanganate, or any of the low regulatory priority drugs, including many of the common fish culture chemicals that are used, like ice, salt, and so forth. So what does this mean for aquaculture? Well, first it means that hatcheries and fish farms must work with a veterinarian licensed in the state of operations. This is the only way that you can access antibiotics, either by prescription or a veterinary feed directive. It also means that extra label use of VFD drugs is now possible and may be warranted in some cases. Previously, medicated feed couldn't be prescribed for anything that wasn't explicitly included on the label. But FDA recognized that there really aren't very many approved drug claims for minor species like fish. And so they have provided this flexibility to veterinarians, allowing them to issue VFD drugs to help sick fish that would otherwise not have any treatment options. So it's good that we have this additional flexibility. For a prescription or a VFD to be valid in any context, it must be issued in the context of a valid vet-client-patient relationship, or a VCPR. And I'll talk a little bit more about what that really means. All VCPRs must include three major elements. The veterinarian has assumed responsibility for the care of the animals, they have sufficient knowledge of the animal to properly diagnose the disease, and they have to make themselves available for follow-up in the case of an adverse reaction or in case the treatment didn't work. This is the federal VCPR definition, and it's not especially detailed in defining any of these three elements, except that it specifies the veterinarian must have recently seen and be personally acquainted with the keeping of the animal and the premises where it's kept. State definitions are, for the most part, very similar, and they address the same basic elements of responsibility, medical knowledge, and availability. And I've included the Idaho State VCPR definition here as an example. State definitions can be more detailed. So, for example, this one specifies that medical knowledge cannot be considered sufficient without the veterinarian having seen the animal or the premises within the past 12 months. That's something that's in the Idaho definition that isn't in the federal definition. The Food and Drug Administration has made determinations for each state as to whether the state or federal VCPR definition applies, and that does vary from state to state. For those of you that are listening and are getting very confused and very troubled, don't worry. This is something that any cooperating veterinarian that you work with will know. But it does underscore the importance of communicating early and often with your veterinarian to know the legal requirements, as well as their perspectives on anything else that they would like to see or do as a part of building and maintaining a valid vet-client-patient relationship with you and with your animals. There are a number of other resources that you can use to get the most up-to-date information about legal drug access and use. The Food and Drug Administration is, of course, the definitive source, and they are the authority. But bless them, their website and documents weren't necessarily written with the farmer, fish culturist, or casual reader in mind. The American Fisheries Society Fish Culture section and the Fish Health section, the American Association of Fish Veterinarians, the Fish and Wildlife Services ADAP program, as well as your friendly local veterinarian and fish health professional, these are really great resources that can help you understand and navigate the somewhat complicated waters related to legal and judicious use of drugs in aquaculture. I just want to mention one especially useful resource, and that's the Guide to the Use of Drugs, Biologics, and Other Chemicals in Aquaculture. This is published by the Fish Culture section, and it's one-stop shopping for any and all of the up-to-date information on the proper use of drugs, including antibiotics, biologics or vaccines, disinfectants, and pesticides. It's really written with the fish farmer or the fish culturist in mind, and it's available free of charge on the Fish Culture section website. It also comes with a handy treatment calculator, and I'll show you that next. This is just a screenshot of 2 pages from the treatment calculator. So essentially you enter information about your tanks or your raceways or your ponds on the first page. That's what's shown on the left. And then the spreadsheet auto-calculates the appropriate application rates for formalin. That's what I'm showing here on the right-hand side, as well as all other approved drugs, as well as the most commonly used INAD drugs. The treatment calculator includes calculations for immersion treatments like formalin, medicated feeds, as well as injectables like spawning hormones. So it's a really great resource, and this, as well as the guide document, are both available free on the website. We're approaching the end of the hour, so I'm just going to offer a few concluding remarks before getting into the questions. This presentation is intended to help the aquaculture industry and fisheries professionals to know the risks and know the facts about antibiotic resistance. An ounce of prevention really is greater than a pound of cure. And so we really want to emphasize biosecurity, vaccinations, and other proactive strategies. You should really know what you're treating before you apply a treatment. You should certainly apply treatments in accordance with the law as well as best management practices. Those resources that I just mentioned are a really helpful way to keep tabs on that. Anytime that fish are sick, it's going to represent a loss. Even if treatment is successful and the fish recover, you've lost time and you've spent money on veterinary services, treatment options, operational changes, etc. So whatever you can do to prevent and minimize that loss, that's going to help you in terms of your operation, and it's also going to address risk for antibiotic resistance development at the same time. As I said in the beginning, there's a lot of mis- and disinformation out there about aquaculture, including antibiotic use in aquaculture, and it's important to know the facts and to be able to communicate these to customers and to the public. So, the first fact that I would reiterate, antibiotic resistance is real, but it's not necessarily pervasive in aquaculture. As I shared with you, there's very little evidence of antibiotic resistance in Idaho hatcheries, and potentially this is the same elsewhere. Since publishing or sharing this information with a variety of people, we've heard from other states who have very similar distribution data, and they're interested in examining it the same way that we have to get a handle on whether or not antibiotic resistance is as concerning in their states as they might otherwise have been led to believe. Second, there's no incentive to misuse antibiotics as growth promoters. That's another fact that's important to be aware of. Number one, it's not legal, and number two, it doesn't work. And finally, as an added fact, antibiotics and really all medications used in U.S. aquaculture are subject to strict regulation and veterinary oversight. And that includes information and regulation of withdrawal periods to protect consumers. Nothing gets approved by the Food and Drug Administration without going through an extensive, almost excessively rigorous process to ensure that drugs used in U.S. aquaculture are safe, effective, and will not expose people or the environment to harmful antibiotic residues. So keep these facts in mind, and hopefully they will help you to not only be aware of the risks, but also to communicate the risks or lack thereof to the public. With that, I'll thank you for your participation in this webinar, and I'm going to check to see if there are any questions now. It looks like there might be one, but I'll mention that if you have questions later, feel free to contact me by the phone or email address that's listed here.